This is TransCoRE, the Translational Success in Corneal Regenerative Medicine project. Here is a normal cornea. It is like a transparent window and allows clear vision. When diseased, it becomes opacified, leading to blindness. The only currently available treatment is corneal transplantation, which restores clarity and vision. However, the supply in corneal grafts is limited and growing scarcer with the aging population. Globally, 12.7 million people are waiting for a corneal transplant, and there is currently only one donor cornea available for every 70 patients who need it. To address this limited supply, researchers have been working on regenerative medicine alternatives such as cell, tissue engineering, gene, and drug-based therapies. However, despite decades of research, few of these have successfully crossed the valley of death of translation from bench to bedside. Furthermore, despite regulatory approval of some of these therapies, clinical uptake by end users, accessibility, and societal impact, especially in underserviced areas, are still an issue. In practice today, corneal transplantation remains the gold standard treatment for corneal blindness. There is therefore a clear need to accelerate the clinical translation of emerging corneal regenerative therapies to address the ever-growing global burden of corneal blindness. One of the problems in doing so is that there is currently no objective way to determine the translational potential of promising emerging corneal regenerative therapies. This is essential both for decisions regarding allocation of resources, efforts, and time to develop a given therapy, and for building a customized strategy to increase the translational potential of a therapy and thus accelerate its progress along the pipeline. Our solution is to develop a translational potential assessment and improvement tool. Our ambition is to make safe and effective corneal regenerative alternatives available to cornea-blind patients in need faster. This tool will directly help cornea researchers developing such therapies, but also indirectly patients living with corneal blindness and their eye care providers. It will also be of direct relevance to funding entities and industry who must make resource allocation decisions early in the development process. Within the scope of this Capstone project, our objectives are to determine the current state of translation of cornea regenerative therapies, including rate and speed of translation, bottlenecks, and determining factors, develop a low-fidelity prototype of a translational potential assessment tool for these therapies, and conduct end-user testing to collect feedback on concept, content, design, and usability. We will proceed first with a literature-based analysis of the state of translation in this field, where we will identify promising cornea regenerative therapies in animal preclinical development published between 1980 and 2010. We will then track their translational progress over 15 years and compare factors differentiating successfully and unsuccessfully translated technologies to elicit those that could be included in our tool. This data will be used to develop a low-fidelity prototype of the translational potential assessment tool, and with the prototype in hand, we will approach a small group of cornea researchers to test the prototype and elicit feedback. Our team is composed of myself and 4 research associates, Marianne Gingras, Dr. Alison Bernstein, Lorena Mija, and Miriam Belesh. We have Alexandra Marzifkin as consulting information specialists, and PAC members Drs. Mark Lesk and Kelly McShane, as well as Joseph Farabach as TRP advisor. That's all from me. Thanks for listening, and please get in touch if you want to learn more about this project or my research work in general.